Both. They were really good. And now I'd like to introduce our final speaker of the day, our early career keynote speaker, and it's really with great pleasure that I'm introducing him. He is a keynote invited talk because he was the winner of the 2021 RMS Early Career Award, our very first cohort of that award. It's Kevin Whitley. He is based at Newcastle University, but his postdoctoral work has been admirably collaborative, split between Newcastle and Delft University of Technology. And he's now going to present the fruits of this work with a talk entitled Combining Nanofabrication, Microfluidics and Microscopy to Investigate the Dynamics and Functions of the Bacterial Cytoskeleton in Bacterial Cell Division. So over to you, Kevin, if you could share your screen.
There we are, right?
Yep, perfect.
Right. Yeah, so thanks so much for that introduction. I mean, it's wonderful. I'm really honored to be given this award. So to start out, I'd like to just give you the kind of personal background of how I got involved in this project. So I spent my PhD doing single molecule biophysics in vitro. So I was using optical tweezers to play with DNA and such, right? And so for my postdoc, I wanted to take this a step further and say, okay, let's look at how single molecules are going to behave in living cells, right? And I was especially interested in looking at bacterial cell division, which I'll mention why in a moment. And so I was looking around for postdocs, and I found this position that was being advertised between these two groups, K. Stecker, Delft, and Seamus in Newcastle, and the goal of which was to find a postdoc that worked between both groups and combine the expertise of both of them, so like nanofab and advanced microscopy, to tackle this subject. And so, of course, I couldn't pass that up. That was the subject I wanted to do, and involved this heavy method development aspect and new microscopy methods. So here I am. So advanced slide. Go. There we go. All right. So why are we interested in studying bacterial cell division? Well, there's basically two main reasons, at least for me, why I think this is important. One of which is it's a major target for current and probably future antibiotics, so think penicillin, because antimicrobial resistance is, of course, on the rise and is predicted, if nothing is done, to outpace the number of deaths from other causes within a few decades. So we urgently need new antibiotics. And from another more, I guess you could say, philosophical aspect or basic biology, there's this effort to see if we can build a cell from the bottom up, basically just take some whatever minimal components we can, some DNA, some proteins, some lipids, and see if we can get that to grow like a living cell and even to divide and reproduce. And so, of course, we would like to understand how relatively simple organisms like bacteria do this so that we can inform that. And so, for a lot of you here, I guess many of you are not biologists, right? Or microbiologists. So you might think, why are we interested in bacterial division in the first place? Like, isn't this a solved problem, basically? But it turns out it's actually a really complicated problem. So here's a model organism that I'm going to be showing throughout this talk, Bacillus subtilis. It's a rod-shaped organism surrounded by this thick cell wall. But so the physical problems that this cell has to solve in order to divide are actually pretty remarkable. I mean, it's under pretty high turgor pressure pushing outward. And it's surrounded by this rigid mesh of peptidoglycan, which is a polymer surrounding the cell. And so it's against this pressure and against all of this rigid polymer that it has to continually remodel and build further inward in order to divide. And at a molecular level, this is also a very complicated biophysical puzzle that needs to be solved, because there's a collection of a bunch of proteins that are going to work together and cooperate to build this cell wall further inward, this cross wall or septum. But each protein is on the order of, say, 5 to 10 nanometers in size, thereabouts, and they have to work together to build this ordered septum over a distance of about a micron, so it's about 100 times larger than they are. And so the collection of all proteins that are involved in doing division is called the divisome. So, I mean, fun fact, anything in biology is like 10 times cooler sounding if you add "ome" to the end of it, and so that's why this term exists. Anyway, but so divisome proteins vary a lot from species to species, and there's still tons of work that still needs to be done to understand the functions of each of these and such. But thankfully, there are a few shared components across most bacteria. one of which is the synthases that actually build the cell wall, the PG synthases, because of course they are going to need to be there. But another is this protein FtsZ. And so this is a cytoskeletal protein, so you could think like microtubules in eukaryotes. It's actually a homologue of that. And this protein is important because it's almost universally conserved throughout bacteria and essential for division in almost all. There's a few exceptions, but by and large, it's very widely conserved and essential. And so because of that, it has been the subject of intense research over the past 3-ish decades or so. But I'm going to skip ahead to 2017 to some results that our labs contributed to prior to my arrival. So 7 groups were working together actually on this subject in different species. And they looked and found that FtsZ, even though it's the skeleton of the cell, it's highly dynamic. It actually moves circumferentially around the cell, and in doing so likely guides the synthases to build the cell wall. But it's an unusual kind of motion. So I'll play this animation. It's called treadmilling. And so this is where the filament center of mass does move. There's this collective motion, but actually each individual monomer doesn't. Each one just kind of binds onto one end, sits there for a while, and then eventually falls off the other end. But the filament itself does move. And so this is called treadmilling. But despite these advances, it's still unclear basically what the physiological role of FtsZ treadmilling in the cell is, despite how important it is. So I'm just going to show you what the conclusions are right from the start, and then we'll go into especially more the microscopy and the method development, because of course this is a microscopy congress, right? So anyway, with FtsZ, we found that it actually has 2 roles, one of which is to basically set up the FtsZ ring, the division ring, and prepare it for division, and afterward to guide the synthases. But there is a point at which it does become non-essential, so you can just stop all FtsZ motion, and cells are still going to keep dividing, which does have some implications for antibiotic design as well as for informing the bottom-up biology. Right. So we wanted to look at FtsZ treadmilling across the whole cell cycle to see really what its point is. But of course, that raised the question: what are the stages of division? How does division happen in bacteria? So we looked at this in terms of the large macroscopic structures in the cell. So this is a video here of Bacillus subtilis cells. There's, I guess, a couple of them in this field of view. Well, 1, then 2. Where FtsZ is labeled with a fluorescent protein. So we are just looking at the larger scale changes in the cell over time. So we quantified this with some tracking software as well as some MATLAB software that we wrote to quantify these larger scale changes in the cell, just its diameter and its thickness. So here's just some traces showing that, of course, the black lines are representative, whereas the gray dotted lines are many others, diameter and thickness over time. And so from these time traces, we can identify 3 stages for the cell division. One of which is what we call the nascent ring stage, where it's still not even really a ring at this point. It's kind of a diffuse collection of filaments, very blob-like, that you can see in the video. But then after that, this condenses into a tight, dense Z ring, which we call the mature ring, before it has actually begun constricting. And then after that, it is now constricting. The diameter is decreasing. So, okay, we wanted to look at FtsZ treadmilling at each of these stages to see what's going on. But doing, molecular level imaging in bacteria is difficult for a few reasons. One of which, little known fact, bacteria are small, okay? And I mean, they're much smaller than eukaryotic cells, which is often, I think, maybe not quite as appreciated as it could be. But they're not that much bigger than the diffraction limit of visible light, right? And so if you just put some single molecule in there and you image it, well, I mean, you're going to get a lot of signal overlap if you have more than one, especially if you're looking along the short axis of the cell, which makes this actually pretty difficult to image. Another fact that complicates this is that a lot of bacterial cells are rod-shaped, especially most model species and a lot of human pathogens are rod-shaped. And so when you image them on a microscope, they lie flat, so they're horizontal, and the division ring, you view it side on. So it's projected into a line, essentially, on the cover slip. And you see substantial signal overlap. And you can get around that with using total internal reflection fluorescence, where you only illuminate the bottom, say, 100 nanometers or so of the cell. So you remove a lot of these out of focus filaments, but then you also lose the context of the entire cell. So, I mean, this is what you get from that. You can get quantitative information out of it, although it is actually very difficult. And I can say from analyzing some of these images, it is really difficult to get information out of that. So then, wouldn't it be great if instead of having the cells lying flat on the microscope cover slip, you just stand them upright? You just put them in tiny little holes that are the shape of bacteria, and then you have the division ring in a single imaging plane, so you have higher resolution and no signal overlap. And so what we do is we use nanofabrication techniques in a clean room, e-beam lithography and such, to make micro pillars on a silicon wafer to use as a mold, and use those as a mold, pour on some molten agarose, let that set, and then you have some bacteria-sized holes that you can stick the bacteria into, so they're all stood up, and you can see them all in, a nice, neat array in your, field of view. And so in doing so, we can see FtsZ moving around the circumference of the cell in pretty high resolution without even messing with any, fancy fluorophores or anything like that. Actually, I'm going to replay this because it's a cool video. Wait, hey, you're supposed to play. Anyway, so we are now calling this technique VerCINI, for vertical cell illumination by nanostructured immobilization. And even though this technique is really good resolution, we found that it's actually not even quite enough for looking at nascent Z rings because they're so dispersed and so weak in signal that it's really hard to see anything. So we had to adapt and come up with some image processing methods to fix this. And this video really doesn't like to replay, so I'm just going to have to do this manually. So anyway, we denoise the images first, and then we use an explicit model to subtract out the cytoplasmic background. And so in doing so, with VerCINI, with this image processing, we can get the highest sensitivity FtsZ treadmilling that has been done to date. And how we analyze these videos is we look at treadmilling for some time. We fit the cell to a circle, and we produce a chymograph, which is radial chymograph. So this goes from 0 to 360 degrees around the cell, and then a second time around to 720 degrees. So it's plotted side by side. And we do that just so we can see this boundary between 0 and 360 degrees much more easily. So how to interpret these chymographs? Imagine if you had, say, a stationary particle that's just stuck at 180 degrees around the ring. So over time, if it just sits there, you're going to have this vertical line at 180 degrees. Whereas if you had directed motion, if you had something going from 0 to 360 degrees around, you're going to end up with some diagonal line. And so what you see in this example chymograph here is a lot of diagonal lines moving in both directions because FtsZ is treadmilling bidirectionally around the cell. Right. So then coming back to this point, we wanted to look at how this treadmilling behavior is happening across all these different stages of division that I mentioned before. Nascent, mature, and constricting. So we've done this for a bunch of different cells now. So the mature and constricting rings look pretty similar to the example I showed you before with this kind of almost lattice-looking diagonal lines crisscrossing and all that, consistent treadmilling. Nascent rings are much more sparse, and they have a lot of filaments that aren't really moving much. They're just kind of sitting there. So, of course, we've done this for many cells, and this is just a summary in the violin plot saying what I just said. Nascent rings, a lot of really static filaments. Some are moving variable speed. Mature and constricting rings, very consistent motion, actually. Right. So that was pretty cool, but we wanted to take this a step further. And we wanted to use specific antibiotics to perturb the cell and see what's going to happen to the system in that case. But the problem is, with this original implementation of the method, we have this block of agarose sitting on top of the cells. And so it's not very easy to control the chemical environment because it has to diffuse through this porous medium. So the idea then is if we just invert the system and we make it so the holes are open on top and we put the whole thing into a microfluidic chamber, then we can more accurately control the chemical environment and flush over different nutrients or perturbatives like antibiotics. So this works basically the same way. We have our silicon pillars as a master, pour on some PDMS. Some of you might know it's commonly used for microfluidics. And then peel that off, and we have a cover glass with PDMS on it. And then we get the cells into the holes, which it turns out is not a trivial matter. We actually centrifuge them into the holes by spotting them on top of the holes and sticking the whole thing into a centrifuge and just spinning them in. But so at the end of this, we have a microscope cover slip that has the cells loaded into the holes that are facing up, right? So what we want to do then is put this into a microfluidic chamber so that we can actually flush over perturbatives and such. So from this, I took a page from my single molecule background and made some low-tech flow chamber. And so this is a microscope cover slide that has some holes drilled in it, some pipette tips inserted in and epoxied in place, and a flow channel that's made out of a piece of double-sided tape, which is about 100 microns in thickness or something like that. And I found that actually the brand of tape really matters. I use duct tape. I found out that Scotch doesn't really work, doesn't stick to PDMS super well. Anyway, minor point. But so we assemble this whole thing, and we have this nice low-tech, low-cost microfluidic chamber over which we can flow stuff and affect the cells. And we're now calling this technique VerCINI for microfluidic VerCINI, remember? Okay, so we set out to test this method as well as to gain some biological insight by using this antibiotic called PC19. So this compound specifically inhibits FtsZ. And how it does that is it actually binds to it and it prevents it from depolymerizing. So it's unable to move then. It's similar to Taxol for microtubules, if you're aware of that. It's an anti-cancer drug. But it still remains unknown up to this point what the timescale of effect is and if it really works well against rings that are in the middle of constriction, if they've already, started going. So using VerCINI, we record the dynamics of FtsZ treadmilling for a while. This looks the same as the things I showed you before. But then, of course, we flush in this compound and things go out of focus for a few seconds, hence the black band there. But then after this compound hits the cells, within a matter of a couple of seconds, basically, all motion stops. FtsZ treadmilling is halted, and you just see these static filaments afterward. So it is actually a very potent compound, and it works very well against all these stages of constriction that I showed before. So we then wanted to see what's going to happen when we stop FtsZ treadmilling abruptly in rings that are in the middle of trying to condense their ring or trying to constrict. And in this case, we just used horizontal cells because it turned out to be faster and easier, and we didn't need super high resolution for it. So recording the cells growing, dividing for a while, flush in PC19, and you can see this very characteristic aspect of PC19 where FtsZ clumps form when they can't depolymerize anymore. It's a very dynamic process. But so, of course, from these videos, we wanted to try and see what is this doing to the condensation of the Z ring and also to actively constricting rings. And so if we look at a bunch of nascent rings, which of course aren't really rings, it's almost a misnomer there, after PC19 hits, you see basically nothing. I mean, there's no ring or anything that forms, it's just blobs of junk. I mean, they never condense. And I gave a few the benefit of the doubt because they maybe did. But for the most part, none of these rings are able to condense after this compound hits them, telling us that FtsZ does need to be treadmilling in order to condense, probably just to interact with other filaments that are moving around. Then looking at constriction, we wanted to see if this was going to affect how the cells are constricting when we inhibit FtsZ treadmilling. Nascent rings, of course, don't. I mean, they don't even form a ring, so, move past those. For mature rings, we see two different behaviors. Some stop and they don't constrict at all, and some do, actually, even after the compound hits them. And for rings that are in the middle of constriction when this compound arrives, they continue constricting pretty much unaffected. And that's summarized in this stacked bar plot here. And we went a bit further than this to show that actually it's the arrival of the PG synthases that are building the cell wall inward that corresponds to when FtsZ treadmilling becomes non-essential. So when those enzymes arrive, you don't need FtsZ around anymore, apparently, or at least not to move. But this was a surprising finding to us because we, of course, know that FtsZ is still dynamic in constricting rings. It is still moving, definitely. And we know that it does, to some extent, affect the rate of constriction. So we wanted to more sensitively look at rings during constriction when FtsZ treadmilling has been knocked out. So we tracked these cells, we measured their diameters with our MATLAB software over time, right? So we have, this is the diameter of the cells over time, they're constricting, right? And we fit these to a very simple toy model for constriction where all it really assumes is that the rate of cell wall insertion going inward is constant. And so that just begins at time t equals 0 there. And so from this, we can get the rate of constriction as well as the time that it takes to constrict, extrapolating from 0 to the end there. And comparing that from the cells that have active Z treadmilling to those that have it knocked out, those that are treated, the ones that have been treated constrict considerably more slowly. And I mean, after eventually finding this, I could look back to the videos and be like, I guess they are actually slower. Interesting. So it's slower, but the cells are still able to divide even after treatment with this compound. And so then what's going on with this? So our model for this is that FtsZ treadmilling is needed basically to confer long distance order to the PG synthases. Like I said, these synthases are 5, 10 nanometers in size. I mean, they don't have any directional vector telling them where they need to go. But FtsZ is forming these filaments that's able to move. So that might be actually giving them that directionality that they need. But then after you have a septum that's already started, it's already started building one inward, the synthases already have a track to move on, so to speak, and maybe they don't really need FtsZ to guide them anymore. But in the same sense, maybe there's some effect. So if we look at a few cases that I've found, when the cells are treated, sometimes the dividing septa go very strange and off-axis, like diagonal, which may be, an indication of this. This has also been seen in previous SEM images where the septa end up really messed up looking after they've been treated. So the cells are still able to divide. They just end up with a really messed up looking septum, which is probably not very happy. Anyway, so this comes back to the final conclusions for the FtsZ treadmilling stuff. That you do need FtsZ treadmilling to condense the ring. Probably just treadmilling filaments are moving around the circumference and then running into each other. And then other factors are bundling them together and forming this nice dense ring. And then after that, they're probably guiding the synthases by giving them some longer distance order. But of course, crucially, after the septum has begun, you don't really need it anymore, apparently, which also says that, if you're designing antibiotics against FtsZ, which would make sense because it's essential for division, there is a subset of cells this is not really going to be working on, those that have already started division. And also this is helpful for, say, the minimal cell effort, trying to build the cell from the bottom up, that you might not want to use just FtsZ, at least, to divide the cell, because it seems like, at least in bacteria, they need quite a few other components, and that FtsZ itself is not the only thing you need. Right. But so then I have just a few more minutes. I'll just explain kind of how I'm following this up at the moment, because I find it interesting. Right. So I mentioned that these PG synthases are the ones that are moving and doing all the actual work of building the septum. And so that's what I'm trying to do now, is to look at the synthases moving it at single molecule resolution. So I'm doing single particle tracking on these synthases while the cells are confined vertically in using our VerCINI method, and watching these single molecules moving around the septum, probably doing their thing, building the wall. And in this case, we have a single molecule that we're tracking around the septum for close to 5 minutes there in that case. And I'm hoping to get even better data in the future, or at least a lot more that look like this, because these are very nice tracks to see. And you can see a lot of this rich behavior in the distance that it's moving, that there are these direction changes, there's pauses. I mean, there's a lot of rich behavior that we have to look at, which I'm really excited to get deeper into this project. Yeah, so for my last slide here is basically just an advertisement for VerCINI. So you don't have to have rod-shaped cells for this. Any cells with cylindrical symmetry, including streptococcus, like we heard about in the last talk, will fit in these holes that you can image. And you also don't even have to image division proteins with these. We found that you can also image the cell poles with them really well. In fact, because the cells are adjacent to the cover slip, you can use turf, like I mentioned before, to only illuminate the very bottom 100 nanometers or so of the cell. And you can see the poles of the cell in, with high signal to noise ratio. So here's an example of a strain that we got from Henrik Strahl here in Newcastle, which is a chemoreceptor protein. And you can see that in horizontal cells, it's really hard to see much of anything that's going on. But if you orient them vertically, you can see these large chemoreceptor clusters, which are extremely immobile, as it turns out. But we think that you could do quite a lot more with this. So, I mean, get in touch if you're interested. I mean, we are trying to democratize this method. We have this new preprint out, and we're in talks with a company to try and make these silicon wafers that we can actually, distribute to other groups. No proceeds come to us, but yeah, we hope that you'll find this method interesting if you're doing bacteria and such. And so obviously all this is a team effort, and a large team indeed. So I'd like to acknowledge all the people who've contributed to this, both in the Holden Lab and in Kay Stecker's group, now solely based in Seamus's group, but of course, I acknowledge the work that was done, in both groups and everyone that helped out along the way. Special shout out to Callum Jukes there in the X-Men shirt, who is I think it's X-Men anyway, who's a PhD student in Seamus's group who contributed quite a bit to what I presented today. And one last thing is I'm now looking for PI level positions. So, hey, if you're a department that is looking for a microscopist, get in touch. I'd love to hear from you. So with that, I will finish. Thank you all for listening. Thanks.
Thanks, Kevin. That was a great talk, and I enjoyed the little advert for yourself at the end there as well. I hope someone comes back to you. Please, people, feel free to pop a question in the chat. I'm sure there's lots of questions coming. In the meantime, I was wondering a bit about the distance from the objective to the that actual ring. When you're sitting them upright, it must affect the imaging a little bit, or the focus working distance of the objective that you need.
Well, at least in the case of the original VerCINI technique, it's about 1 or 2 microns off the surface. So that one's not so bad. We use a 100x turf objective for that one. And I mean, we have pretty high resolution, as you can see. With the other method, mu VerCINI, where we have the PDMS layer on top of the cover slip, that makes it much more difficult. And we found that with the 100x turf objective, we can't actually image high enough because the working distance limits it. So in that case, we use a silicone oil immersion objective, which is for imaging into deep tissues, I guess, is the reason why it was made. But that enables us to get much higher working distance to image all the way up there.
Yeah, and do you get much signal loss having to get that extra distance?
It doesn't seem like it. I haven't quantified the loss of signal, but yeah, it seems pretty similar, at least in both methods. So, yeah.
And how about the imaging pores when you're treating the cells and they're extending? Because we could see in the horizontal phase that they were extending quite rapidly. Do they extend out of the pores? How does that work?
Yeah, they do actually. And so if you're doing this in, say, fast growth conditions, sometimes you have to do the imaging a bit quickly because they will start to grow out over time. And that's one of the reasons actually we're doing the constriction work in horizontal cells, is because we did find that cells were growing out of the holes. Actually, I have a slide for this. There we go. So we were imaging constriction in these open top microholes initially, but the problem we had was that over time, a lot of these cells are just popping out of the holes. They are just growing out over time. So this is probably a surmountable issue, but we haven't devoted enough time yet to trying to find a way to really glue the cells into the holes to make sure they don't grow out.
Sure. Okay, I asked a predictable question there. I've also got a question from Liam. He's asking, how picky do you need to be when tagging the FtsZ to retain full functionality, and are there any particular tags to avoid?
So with FtsZ, it is just a tricky protein to tag in general because it is so essential and it interacts with a lot of other things. So this strain we're using dates back, over 20 years actually, because it's an unusually robust FtsZ fusion protein that is actually fully labeled. You can also express this at dilute levels, and in that case, you can use a number of other labels like neon green and things like that. But we've chosen to use this FtsZ GFP strain mostly because it doesn't really perturb the cells as much as most other tags do.
Okay. And I've had another question about the inhibition. Have you tried reversing it or removing the inhibitor, and do you see the rotation movement begin again?
I have, yeah. Early on, I tried doing those experiments, these kind of wash out experiments, as I called them. And yeah, I think I was able to see some restarting of treadmilling, but honestly, it's really difficult to see because I think this particular compound does stick fairly well into the FtsZ. So, I have to try and image. What I was doing is imaging, for a while, waiting 10 minutes, coming back, trying to image it again, because I think FtsZ does restart treadmilling after you wash this out, but it probably takes a little while to really get to that point.
Okay, it sounds like quite a robust inhibitor and the timescales that it works.
Yeah.
Yeah. Do you think the agarose as well maybe absorbs some of it? I don't know how embedding in agarose could.
Well, at least in the case of the when treating with the compound, there was no agarose in that case. That was with PDMS. We have looked at a few other inhibitors, though. There's one that had been developed called PC58, another kind of experimental antibiotic. That one works the other way by preventing FtsZ from polymerizing in the first case. So we've also looked at that one. Unfortunately, it didn't quite have the shocking effect that we were hoping, because the constricting rings did not completely dissolve when we treat with this compound. They're pretty well stuck together, unfortunately.
Sure. Okay, well, thanks so much for answering all those questions and this fantastic talk. Thanks for also applying as our first medal winner. I'll let you off the hook now and hand back over to Liam, who's going to kick off the AGM section of the meeting.
Super, thank you, Becky. Yeah, that was a great talk, Kevin. Thank you very much. And thank you to our other speakers in that session as well. So yeah, this next part of the meeting involves the